Hello children, we were studying structure and germination of seeds. In the previous lecture we studied about various conditions necessary for germination to take place and different experimental setups. Today we will start with types of germination. Before that, I would like to tell you the scope of syllabus of this particular unit, so that while studying you stick to the scope of syllabus. So the first thing that you need to focus on is structure and germination of bean seed and maize grain. Only bean seed and maize grain. Difference between monocot and dicot seeds. Difference between hypogeal and epigeal germination that we will be doing today. And conditions for seed germination supported by experiments. So these many things are there that we must cover in this unit. So let's start with types of germination. On the basis of behavior of cotyledon, germination is of two types, epigeal and hypogeal. Now we will never find both epicotyle and hypocotyle of seed elongate together. Now epicotyle and hypocotyle, children, I hope you are understanding that what I am talking about. We have discussed embryo axis in detail. When I say hypocotyle, that means I am talking about the region beneath the cotyledon and just above the root, right? And so when hypocotyle elongates, then what happens? The cotyledons, they are lifted above the ground. So cotyledons will be coming above the ground, we can see them. This type of germination is called epigeal germination, and generally we see epigeal germination happening in dicotyledonous seeds, for example bean. Then if epicotyle region elongates, then the cotyledons will remain underground, or they will just touch the surface of the soil. They will not be lifted above the ground. So if epicotyle elongation takes place, then this type of germination is called hypogeal germination, and it usually occurs in monocotyledonous seeds. Example is maize. Now see these diagrams, children. These diagrams are from your textbook. Alright, so see the first diagram, that is germination of bean, an example of epigeal germination. See children, during the process of seed germination, first of all the seed will absorb a lot of water. This we already know. Water absorption will make the seed swell, and ultimately the seed coat will rupture. Once the seed coat ruptures, then the radicle emerges out. In the third sequential diagram, children, you can see that how the radicle has emerged and it has grown and formed the primary root. In the fourth diagram, you see the hypocotyle elongation takes place. So the hypocotyle elongates, it straightens, and cotyledons are pulled into the air. Cotyledons are lifted above the ground. You can see that in the fourth and fifth sequential diagram. Okay. Now these cotyledons, they give rise to the cotyledonary leaves. All right. After some time, what will happen, that these cotyledonary leaves, they will shrivel and will be shed by the plant, and young leaves will unfold and they will take up the activity of preparation of food. Alright, so what happens in case of epigeal germination? The seed coat ruptures, radicle emerges, primary root comes out, and hypocotyle elongates. Because of hypocotyle elongation, the cotyledons, they are lifted above the ground and they give rise to the cotyledonary leaves. The leaves, they wither, they shrivel, they die. In the last diagram, in the sixth final diagram, you see the dried or the shriveled cotyledons are, you can observe, and young leaves have emerged, that also you can see. Alright, so this is what happens in case of epigeal germination. Now children, focus on the second diagram, that is germination of maize, an example of hypogeal germination. Here, as I said, the cotyledons will remain under the soil or just on the ground during seed germination, and rapid elongation of epicotyle takes place. Alright, so again the seed coat will rupture, the radicle will come out, giving rise to the primary root, and here we all know that both radicle and plumule are protected by their sheaths, that is your coleoptile and coleorhiza. So they will come out only after piercing through the coleorhiza and coleoptile. Alright, so plumule will emerge only after piercing through the coleoptile. Radicle pierces the coleorhiza, grows downward, and it is giving rise to the primary root system. In the third sequential diagram you can see. Then the primary roots, after some time they will perish. Alright, and they will be replaced by the fibrous root system. That is a characteristic. So it has fibrous roots. So the primary root will perish, and in place of them fibrous root system will develop. Plumule will pierce through the coleoptile, and it will grow upwards to give rise to the shoot system. Young leaves unfold. Alright, so this is what happens in case of hypogeal germination. I basically focused on germination of bean and maize, right, because I've already told you the scope of syllabus. Apart from this, in your book you will also find one more example of hypogeal germination, that is in the seed of pea. Now since it is hypogeal germination, same thing is happening there. The seed coat will rupture, the radicle will elongate, and it will grow downward in the soil, and epicotyle elongation takes place. Okay, now when the epicotyle elongates very rapidly, then what will happen? The plumule, it becomes free and it comes out, young leaves appear, and they start producing food. And here also the primary roots initially is formed, and later on it is replaced by taproot system. Now children, this diagram of germination of pea is there in your book. I want all of you to go through the description and the diagram once. If you understand these two diagrams properly, you won't face any problem in understanding germination in pea also. Alright. Now, you can tell that what is the basic difference between epigeal germination and hypogeal germination. See children, in case of epigeal germination, hypocotyle elongates, and in case of hypogeal germination, epicotyle elongates. In case of epigeal germination, the first diagram, cotyledons come above the soil, they are lifted above the ground, and in case of hypogeal germination, the cotyledons, they remain below the soil, or they will just come at the surface of the soil, they are never lifted above the ground. So these are the two basic differences, right? Now moving on to viviparous germination. Now children, you have studied the term vivipary while studying classification of animals. You know that what is vivipary? When animals, they reproduce by giving birth to their young ones. Now here also, in case of viviparous germination, what happens? That the seed starts germinating while it is intact and while it is in the fruit. Okay, so the seed germination begins inside the fruit when the fruit is intact with the parent plant, and when the seedling stage comes, then the seedling is dropped in the ground, it fixes, it anchors itself with the help of the initial roots, and the seedling then develops into a plant. That is why it is known as viviparous germination, because the seed is not falling and getting germinated. Instead, the seedling falls down, and that seedling, it anchors itself to the muddy soil, and then it develops into a plant. Alright, now this is a very special condition, and viviparous germination is usually found there are some examples like Rhizophora, Cereops, Soneraceae, etc. So generally we will find this type of germination happening in the trees or plants which are found near the seashore. Alright, like Rhizophora, this is what? This is a true mangrove plant, a tree, okay. They are known as true mangroves, and mangroves are, they grow in dense forests near the coastal regions, in salt marshes. So generally this vivipary will be found in such plants which grow in saline marshes or along the seashore. Alright, so as I told you, here also it is written, children, the seeds of plants growing along the seashore or in saline marshes, they show a special type of germination called vivipary. The seed germinates inside the fruit while it is still attached to the parent plant, and the seedling emerges, it gets detached from the parent plant, gets fixed in the muddy soil, it gets anchored through the developing roots, and then this seedling starts developing. Here we have the diagram of vivipary. You can see, children, that how the radical has emerged and it is still intact, it is attached with the fruit, and the fruit is there in the parent plant. Now when the seedling is formed, it falls down, it gets attached, the roots, they get fixed in the muddy soil, and then the seedling develops. Alright, so there are two diagrams, right? So children, with this we come to an end to the types of germination, and I will do a proper revision of this chapter, okay. So in the next lecture we will revise the entire chapter, and I will be covering this hypogeal germination in the seeds of pea there. So please study that, and tell me in case you have any confusion anywhere in the chapter. All right, thank you.